All right. Well, thank you for coming. This is a IAS Distinguished Lecture and co-organized by Division of Life Science. So today, my great pleasure to introduce Professor Ali ERTÜRK. And Ali is coming from Germany. Currently, he's a full professor at LMU Munich. He's also the director of Institute for Intelligent Biotechnologies. That's also in Munich. And he has another role as a CEO and founder of a company called the Deep Fiction. Ali's education, he received bachelor degree in Bilken University, I believe that's in Turkey, and then doctorate degree at Max Planck in Munich, Germany, postdoctoral training at Genentech, that's in South San Francisco in US. Ali obviously have a very distinguished career, mostly in Munich. He's known to discover the tissue clearing technology, and this is at the time, including my own career, have very strong impact. You'll actually be able to see, specific protein, mostly through immunolabeling in the organ tissue, and I think we're going to hear whole mouse body as well. And so his research focus now is to use this technology, tissue clearing driven cell level imaging of human organs and mouse body for the biomedical innovation. So let's give a round of applause to welcome Ali for a special seminar.
Thanks very much for invitation and introduction. It's a pleasure to be here. First time in this institute, and good to see all your facilities and learn a bit about ongoing research. And today I would like to talk about some of the technologies that we have been developing in the last couple of years and how we apply it to understand the biology in an unbiased way at cell level and to develop also therapeutics at cell level, those that we want, that they achieve cell level accuracy. So, as you know, we are an organism of interconnected systems. We are wired with vessels, nerves, muscles, other systems end to end, right? Therefore, clearly, a disease affecting one part of the body will affect the rest. I think cancer metastasis is a very clear example of it, right? So we cannot just say we will focus on liver for liver cancer. It will go around and cause a lot of damage or invasion of other tissues. So ideally, we need to look at the big picture to understand all the cellular events, entire body, and try to find, because then we can make sure that we treat diseases end to end, right? That should be a really ideal way to do it. But we couldn't make it so far in this way because in the lab, we focus on organs. We take out, we section, and we look at this tiny piece of tissue, but we are ignoring the rest. That's a problem because then we are biased. We are not seeing what's happening in the rest. A clear problem for that because imaging modalities such as MRI, PET, they don't have resolution really to look at the cells and molecules, right? You can see gross anatomy, but you cannot really look at cells and molecules using this kind of whole body imaging technologies. So what we need an imaging technology that enables us to look at the whole biology at cell level, such as whole mouse bodies that we work often with. To enable this, we develop so-called tissue clearing technologies where we convert biological tissues into transparent structures. As you can see here, whole mouse body transformed into transparent glass-like structure. You can think this converting milk into water. We can see through and scan through with the laser scanning microscopy. So now have a look at this. We have whole mouse, transparent in the solution. You don't see it. We take it out. You can see it more clearly because in the air, it looks like glass. When we immerse into solution, it disappears because refractive indices are the same throughout the solution and the mouse body. That's how we make it transparent. Now, when we scan it with the lasers, laser scanning light chain microscopy, we will be able to eliminate everything inside and start collecting cell-level information without sectioning. So these are fluorescent microscopy scans. We obtain hundreds of thousands of images from one animal. We scan several billions of cells in one sample. And these are maybe 15 terabytes of data alone for one mouse. So it's almost impossible, or it's actually impossible to analyze it by hand. Of course, having nice images alone is not sufficient. It's good to look at it, but to understand biology, we need to analyze them carefully and well. For that, we have been developing deep learning algorithms in the last 10 years or so, for example, to analyze cancer metastasis and drug targeting at cell level, to analyze vasculature and neuronal activity throughout the organs and whole bodies at cell level, as well as to map individual cells in 3D in entire human organs. So our core technology, I think, is clear to everyone, right? Clear what we do? Transform biological tissues into transparent structures, in particular whole mouse bodies, that we can see everything at cell level, such as diseases, cancer metastasis. And if we give a drug, we can also see it at cell level. So that's the core. I think that will stick with you, hopefully. Now I will show you some applications of this technology what I mean with disease characterization and drug development. So recently, we applied this into obesity model and study the changes of the systems, biological systems, in fat animals versus lean animals. And we already established tissue clearing long time ago, more than 10 years. We do this at whole mouse level. But we have been developing, as I mentioned, the algorithms to analyze the systems. We developed so-called mouse mapper, which is on bioRxiv. We can check. It's just a network, combination of network called deep network ensembles. There are different networks. One is analyzing the nervous system, other one is immune system, other one then combines this into tissue and organ mapping, registration, and then gives us output. So now if we look at whole mouse body, fat versus lean on the left, you can see we're looking in green the nervous system. This is a transgenic animal, UCLH1, labeling some subset of neurons, axons. You can see actually already some abnormalities when you look at the whole mouse body nervous system. You can see the curling of axons here, for example. Let's zoom in, you can see they're here compared to more straight axons in the normal case, but also we see degeneration of these axonal bundles in high-fat case compared to a standard case. There is this decrease of bundling. So these nerves, they're actually part of trigeminal nerve innervating olfactory stem whiskers. You can see, doing such analysis now, we look at entire impact of a disease like obesity, what it does in the whole body, and we see degeneration of the nerves going to the face. And actually, if you look at the clinical reports, there are also obesity or diabetics, people suffering from sensory motor function. So having unbiased view identifies as the location of changes. That's great, right? Imaging-wise, we identified. But that doesn't tell us much about what's really happening. So what do we need to do now? So we found this region of interest, some changes. What is next? We need to do some molecular analysis, right? We have to do some omics. We have to dive into the molecular underpinning. So for that, we developed a technology called DiscoMS. So after tissue clearing, imaging, and identifying regions, you can see here in cancer model, some of the cancers are targeted by a given drug. They are in white. Some of them are untargeted. We can isolate them and then bring them to mass spec, run proteomics using robotics technologies that we develop. So that's quite powerful because we are unbiased in checking the whole system, finding regions of interest for changing. As I said, this could be very tiny metastasis or could be first plaques appearing in the Alzheimer's mouse brain, or first vascular changes in another dementia model. You identify regions of changes, and now take it out and run proteomics in this animal model where we study obesity. We run the proteomics and identified a lot of proteins as expected, also axonal remodeling, degeneration, and inflammation. So that's really interesting because now we have some molecular information where we can manipulate the system, for example, to rescue this phenotype. So, of course, working with mouse models, great, but always there's translation issue, right? How that reflects what's happening in the human tissue. So therefore, we also started applying these technologies into human tissue, large human tissues. We rendered intact human organs transparent, so a very ambitious way, whole human kidney rendered transparent. And we scan it again with laser scanning glycemic microscopy, and we revealed 200 billion cells in 3D space. You can see here actually there's dye labeling of large vessels, small vessels, as well as glomeruli. You will see some pathology. The green shows the capillaries. On the right, there's a bit less. There's some problem at the tissue level. But if you deep dive, you have this all information of glomeruli cell by cell reconstructed, and how the vascularization happening around this glomeruli, and if there is any pathology related to this at whole human organ level. Again, we're looking at 200 billion cells. So that's very, very, let's say, unbiased way of seeing the things and going from there to the biology at the molecular level. Another example, we rendered whole human eye, also transparent, scanning with the light microscopy, seeing large samples, retina, iris, but also if you go behind the eye, you can see the nerves and immune cells, individual immune cells are visible. So we want to do these studies in complete biological systems. That's actually, for me, a core ambition, that we look at the whole biology intact and go from there into details. And studying human diseases, we were interested in cardiovascular diseases to apply, as still is one of the #1 killers in the world. And we rendered whole human heart transparent and then scanned it. Of course, one thing you have to keep in mind, not to have this question later, this works only in the fixed tissue, right? So tissue is dead. So this is a heart coming from someone who died. So donated their organ, we take it and study it. The same with mouse. So rendering whole human heart transparent and then scanning through. Now we will dive through aorta. You will see these yellow gold parts where it shows the plaques, aortic plaques. And throughout the aorta, you can see small ones, large ones, and there are regions without plaques. So being able to do this, now we can use the robot and isolate, some small plaques nearby. There's no plaque and large plaque. Try to get a molecular progression of plaques from nothing to real big formation. And we identify quite a few proteins that is implicated in cardiovascular diseases, coronary artery disease, but also on the bottom you see quite a few new proteins that has been identified using this study. I think that's also pretty cool. You see both already known things, means your technology is working, and also reveals now new information. So the cases are, of course, endless. You can apply this technology almost any disease. I assume you already think application in your projects, right? So this technology also reveals, very interestingly, new anatomical information. If you studied medicine, you know that this anatomy books has been written some 50 years ago, and since then they haven't changed because everything about anatomy already is there. Is it true? No, of course. not, because anatomy, of course, gross scale, yes, but smaller scale, there are surprises. So while we have been doing these experiments, what we identified actually, there are short vascular connections between skull marrow and meningeal surface of the brain. Very interesting, because we always thought skull marrow or skull, is like a helmet. It's protecting our brain, right? That's the number or only function that we know it is it? Right? That's normal. I think that's we thought. But actually, it's not the only function. We show that there are these connections, and all these immune cells in the bone marrow, these hundreds of thousands, millions of immune cells, they have very quick access to the surface of the brain. And if we do an injury in a model, mouse model, what we see here in red, those are neutrophils, lower part of the brain, but upper part you can see skull. All these immune cells, neutrophils, from skull marrow regions are mobilized into these channels, and they can invade the surface. They can get into the brain. Now you will see some of these channels frozen, and then cells are there in 3D scans identified. So very interesting biology, because neuroinflammation is a big part of neurodegenerative, neuropsychiatric diseases, right? Everyone is interested in. And we always think neuroinflammation is microglia, and it happens in the brain. But it looks like a lot can happen at the borders of the brain, and actually is even probably more problematic, because bone marrow has huge amount of cells, immune cells, and foray into the brain tissue and can get quickly into the brain. That means a significant impact of these cells. Potentially, right? Has to be investigated. And we published this 2018, also along with Matthias Narendorf from Harvard. And then there was a couple of follow-up studies coming up with Johnny Kipnis and Marco Corona. They did some animal models and showed their relevance in stroke, etc., 2021. And then they were all mouse experiments. We were very curious to see whether these connections, new connections we see, also present in human, right? That's very relevant and important. And for that, we again use our tissue clearing technology, isolated or got samples with skull, meninges, and brain, big chunks of brain, head actually, that's head piece. I mean, that's a bit, scary to think how you get such samples. But we get, and then we clear and render transparent, and then now we are able to see these channels again in this setting. Now zooming in, you can see here this connection here called skull meninges connections. You can see this magenta, those are collection of immune cells, and here is the dura, and immediately there is connection. And this is comparatively a diploic vein, so here is 5 times enlarged. So this structure is much smaller, and another important difference is that, of course, the veins in the skull, they're connected to circulation, so there's no start, there's no end, it's just part of the vessel, what you see, but here is a very clear start. It starts at the level of skull marrow, because it's just opening and then brings to the cells to the surface. Very interesting. When we start doing these experiments, we are not immunologists. And those who are experts in immunology, they are like, yeah, okay, this is bone marrow. Skull is bone marrow. It's very normal. There will be a lot of immune cells. So what, right? Good question. So, translation of question is, bone marrow of skull, is it unique? Something to do with brain, or is it just another bone marrow and has similar cells? So to answer this, we isolated several bones in mouse and human, and did proteomics and transcriptomics. And let's start with proteomics. You can see actually skull versus other, and this is the proteomics profile. It looks quite different. You can see these are clustering together, these are clustering, and skull is very different, as well as in PCA plot. Very unexpected, and tells a lot. And this proteome actually, you can check the paper. it has a lot of interesting things. For example, skull marrow has proteins related to brain. So basically the communication while I'm talking, the cells going from skull marrow into the surface of brain and potentially into the brain with diseases is bidirectional. Things are coming from brain and getting into the skull marrow niches, such some of the synaptic proteins. We don't know the function and how that's happening, but very interesting biology happening. Looking at transcriptomics, we see similar things when we compare different bones. We have, for example, here, well, actually you can see one of this protein, NR4A1. Also stainings, you can see highly expressed in the skull marrow versus femur. So we see that immune cells in the skull marrow niches, they're unique and they have potentially unique functions related to brain diseases. And one thing we wanted to also test whether it has something to do with really human diseases. And the work has with intense data on different things, but I want to show this piece of data as well. So we did TSPO PET imaging, okay? So TSPO PET shows inflammation in diseases such as Alzheimer's here in human. You can see increase in brain tissue. Let's look at actually cross-section here. Increase in inflammation, potentially in the microglia. But what is interesting is that in the skull marrow, you can see also there's inflammation, increased inflammation, potentially those cells, macrophages, neutrophils or what, they're also being activated. So it's mirroring, very interesting. And we did this this is here AD cases, more than 50 AD patients, TSPO PET versus control, but also we did TSPO PET imaging in MS, in stroke, and so on. You see similarly that whenever there is brain inflammation, there is increased inflammation in the skull marrow niches. So this data at least tells to me that when we talk about inner inflammation, it's not only about brain, it is also a lot about skull marrow niches. And to me, it's more exciting because it's already very difficult to target things into brain, right? We cannot deliver. But now, maybe we don't have to, right? Maybe we could tackle infiltration of these immune cells outside of the brain. So that could potentially maybe have an impact. To be studied, to be seen. But overall, this interest of skull marrow niches, we are checking it for different diseases. One of them is infectious diseases. Corona impacted everyone, right? And so it took years for us to understand a bit about virus. And one thing we wanted to understand, when there is infection, what happens, which tissues are impacted. And you can see here, scanning the spike throughout the whole mouse body. Again, using our core technology, we identified a lot of tissues, kidney, liver, intestine. All these red signals are showing where the spike protein of virus is binding. Here you can see at cell level all this information, ovarian, testes, a lot also in the head, bone marrow niches, which is again surprising, and all this accumulation in the surface of the brain and the vessels. And something very interesting is that compared to influenza, which is mostly in the liver, but mostly in the lungs, you can see the Wuhan version, the first coronavirus version, impacts again similarly to liver and lungs. But then the versions that are Omicron plus, they impact the whole body. So SARS-CoV-2, that is considered respiratory disease, is not anymore respiratory disease. It targets entire body systems. So, and then for us, of course, maybe digging into at least the part that we know, looking into skull marrow niches, we could very clearly identify the spike protein accumulation in the mouse brain, mouse skull marrow niches, but also going to the surface of the brain. The green that you see is the spike protein of the virus. Very clear, right, where it is. I think no one would expect that. But again, using this technology of unbiased looking, it reveals everything that is happening. And now we are in a good luxury position to choose and work on one. Right? If you want to publish, if you work on several, that will cause confusion. Just focus on one. Here we focus on skull marrow niches. It's not only in mouse, then we get samples. People died from infection, coronavirus. You can see now there's skull samples. In red is the spike protein. A lot of spike protein from someone who died from corona. Yes, maybe expected. I mean, expected that there's spike around, probably in the lungs, but not in the bone marrow, skull bone marrow we revealed. So you can see here now the patient on the first is again, someone died from SARS-CoV-2. Second is skull samples before corona. There was no signal, no magenta signal. And the most interesting data of this manuscript is this one. So this now skull samples, they are coming from people dying today. They die from heart attack, traffic accident, whatever. We check their skull, and there's enormous amount of spike protein still left several years after infection. That's very crazy. And we've seen this in different tissues as well, but clearly spike protein as like a trash accumulate, and it remains years in the body. Some tissues may be okay, but if it's on the surface of the brain, it's not okay. Right? Because we also showed in the manuscript, well, injecting it into the skull marrow, if we check what's happening, it can actually get into the brain and cause increased caspase activation, cell death. So it's not just staying as garbage in the bone marrow niches, but it's somehow slowly getting into the brain. And looking in the proteomics analysis, we see a lot of inflammatory response, neutrophil, extracellular traps, and so on, in the bone marrow, in the meninges, in the brain tissue, all similar pattern of proteome changes. So another thing we wanted to see actually, okay, spike protein is in the body. It seems it lingers, it stays. Does it have effect long term? To say, is it impacting our immune system or other body systems that we will suffer if something happens, or we will age faster, or we will have more problem if there's a disease? That we wanted to test using traumatic brain injury model. We injected some animals spike protein, some control, and applied TBI after 1 month. So it's a bit imitation of infection, but here we use only spike because we really want to isolate its impact. And you see traumatic brain injury, MRI scans. Focusing on the last images, you can see in the spike case, actually, there is significantly more extended ventricles. So basically, having one shot of a small amount of spike injection to the animals, 1 month later they get injured, they're more likely to get larger ventricles and they're more likely to die. And that's actually what's happening. There's quite a few epidemiologic studies coming. People who got corona, they're more likely to suffer from heart attacks or have worse outcome of these disabilities or dying in many cases. So this is long COVID called big umbrella, but something a lot of people are suffering. When we published this paper in December, I received actually hundreds of emails. People are really suffering, and there is no solution. No one is working on it. No one wants to hear about coronavirus. Politically, it's now, everyone is like, don't talk about it. But a lot of people suffer. So now we are trying to find the ways to reduce this spike burden from the body, whether this lingering spike, if we remove, will somehow help them. So running hypothesis is that you can see the spike protein, the virus probably when it was there or generated the spike protein, then virus gone in the long term, but spike protein remains, they can get into brain, cause vascular damage and inflammation. So there's a couple of more examples. We publish on whole mouse imaging, studying diseases such as cancer metastasis. I'm not going into it now. I want to move to maybe the next part, or important part of it, development of therapeutics at cell level. Now we can see diseases at cell level in whole body, tissue by tissue, organ by organ, wherever they are. That's cool, right? This could be infection, such as the virus, could be cancer, this could be vascular damage. Okay, we could isolate, right? We could isolate regions of interest, do proteomics, molecular analysis, great. Now we can see we have molecular information. And now, let's say we design a peptide using AlphaFold, but we want to deliver it targeted, right, to the cells of this. Or we want to do maybe gene editing. When you do gene editing, we need to do it accurate at cell level, right? If you want to help cystic fibrosis, maybe you want to target lung fibroblast, you're going to target lung macrophages next to it. It's not going to work, right? You have to do editing exactly the cell type you want. But if you don't have this resolution, how do you do that? So we work on this and develop a technology called single cell precision nanotechnology, CP Nano, or nanodisco. You can see actually in such a normal way, it's very hard to see things, but zooming in, now we can see drugs here. Those are mRNAs carried by lipid nanoparticles, which are used in mRNA vaccines. We can see down to cell level where they go. You can see here individual cells, and actually the magenta shows the LNP, and cyan color shows the mRNA. And this is 3 days after injection. And that is translated in, most of the mRNA is translated into GFP, therefore there's few LNP is visible, mRNA, the magenta, but more in GFP. So we apply, in this paper you can see several different types of nanocarriers, liposomes, DNA origami, AAVs, we can visualize at cell level and reveal their on targets and off targets. And studying the LNPs that are used in mRNA vaccines, we actually identified a lot of them are accumulating in the heart tissue. And here's mRNA expressing eGFP. Seeing this data, we were a bit like, that cannot be good news. And also, there have been quite a few reports on mRNA vaccines and how they cause myocarditis, pericarditis. I think in China, I don't know, in Hong Kong, was not very common, the mRNA vaccines, but in Europe, in America, a lot of people got it. And quite a few, actually, I heard from several people, either themselves had heart issues, like my heart was beating like that, or they heard from someone, clearly their heart was not normal, some of the people. There is a certain population is impacted, mostly males. But there was no explanation, of course, from Moderna or BioNTech, or probably there was no interest to explain it because then their drug will not sell, and they avoided it. We kind of revealed it using our technology, but also, of course, not only GFP, putting the spike protein. Now we can see actually spike protein also being expressed with all these off-targeted LNPs, lipid nanoparticles, carrying it, and they're translated. And we also going there doing imaging, proteomics, we show that they cause vascular damage. The heart capillaries, they're targeted, and they're damaged. Not good. So, but because they didn't have this technology, something like this, they are using bioluminescence or PET imaging. You can see where it's hugely accumulating in the liver or spleen. Yeah, well, expected. So what? It doesn't tell you much. But to see such details of targeting in different organs, such as heart and meninges in the brain, very important because you don't want your therapeutics, which causes, in spike case, strong inflammation. You don't want it in your heart capillaries or meninges. So that was a bit of an issue. You can see also with the proteomic analysis of this heart capillaries, we see clear immune response, cell response, cell death. And maybe a final related technology I want to show you is that when we start developing this whole mouse Clearing transparency, yes, we can do transparent, but we have to look at the fluorescent signal. That's the base. We scan with the fluorescent scanning light sheet microscopy. Of course, there's GFP. We can look at GFP. Let's say there's this transgenic reporter animal where GFP is expressed in immune cells, CX3CL1, or there's the type 1 EGFP in neurons. Yeah, you can do some. But then there's a few of them, and they're not really 100% replicating, faithfully staining all the cells. And there's hundreds of thousands of proteins we want to look, but there is no transgenic animal. Ideally, it would be nice to be able to immune stain the whole mouse, but there was no such a technology. How, if you do immunohistochemistry, which I presume most of you do, it's already very difficult to stain 20, 30 micrometer section. How are you going to stain 2 centimeters thick mouse body? Well, actually, we did. We developed a technology called wild disco, which that enabled us to do this. Cardiac perfusion of biochemicals, reagents that makes tissue highly permeabilized, then pushing the antibodies, standard size antibodies to whole body. You can see here, using PGP 9.5, the whole nervous system of a mouse, as complete as so far up to date, is revealed. And this is really amazing. You can even see just from the nervous system, the organs, the outlines, the heart, right, the gut. It's very crazy. So how see all these details now. And you can see now in 3D an example, for example, how the nerves are connecting the interconnected organs, kidney, and all the organs around, and the gut, second brain, a lot of axons, serotonergic connections. And you're looking at the lymphatic system of an intact adult mouse, the complete lymphatic system, which is very critical for any kind of therapy, right? If you inject something, it will immediately suck into the lymph system. Or CAR T cells you will inject, they will travel through lymphatic system. So we can do also double labeling, at least in multi-scale. You can see here lymph vessels together, nerves, again in the gut. Of course, that enables now not only a few things, we can look at any kind of structure and cell type at whole body scale, subtype of immune cells, T cells, part of nervous system. Here, immune cells together, lymphatic vessels. And finally, the nerves with lymphatic system. So with that, I'm to the end. I'm very happy to work very international team here. We are 20 countries and 30 people. Quite a few Chinese students also, as you can see here, also probably seen in the publications. And we are also, as you can imagine, multidisciplinary. I have 1/3 of group is computer scientists, and 10-20% works in omics technologies, and the rest is physics, biochemistry, and biology. With that, I'm happy to, discuss with you your questions.
All right, we have plenty of time for questions. Maybe I'll start with the first one. So for the spike protein that you see in the skull and another part in the body, do you think they're from the virus, a dead virus, or they're shed from the virus? Or you later on mentioned that also messenger vaccine could also have spike protein. Where are they from?
Yeah, good question. Actually, we don't know. We didn't study this in detail. We did a lot of RT-PCR to detect viral RNA, but we couldn't really find much. Actually, we couldn't find any on these long-term samples. So, but we can clearly see the spike protein. It looks like they have very long lifespan somehow. The virus itself, there are some people, they claim there's leftover virus, but that's not very much proven yet, I think, in the body. The vaccine, of course, when it's injected, there's the expression of spike protein, one round, then usually it disappears. So it doesn't last. So if we do on animals, inject the vaccine, we see that spike protein comes and then it's gone. But in infection case, because the amount is so high, the expression, it goes down, but still a lot left over. And that's what we see potentially now with this human tissue.
Can you also maybe quickly describe the evolution of technology? I think initially it was just detergent dissolving lipid, and then you have protein and connective tissue left. Has that technology further improved or changed over time that allow you to do whole body or different organ?
So there are different tissue clearing technologies. You maybe heard Clarity, Cubic, and ours are disco line of clearing. Ours is hydrophobic. That means we remove the water, use alcohol gradients. So the aim is to match the refractive indices of tissue layers with solutions around. And for us, one easy way is to remove the water. Water has very low refractive index compared to protein and other molecules. So basically, they come already very close. And using organic solvents, we then remove some of the lipid structures and make a bit of a decolorization by removing the iron, etc., which deflects the light. Things becomes more and more transparent. That is a bit well established now. There's a few other ways to do it, but a major problem has been, for example, to label with antibodies, how you label such a thick tissue if you want to look at specific things. Therefore, we continue to work on chemistry that permeabilize the tissue. In mouse, it's still doable even at the whole mouse level. As you can see, in human tissue is much more difficult because aging human tissue is a lot of fibers and extracellular matrix proteins. With mouse is 3 months old versus you get tissue from 80 years old dead person. It's a very different level of tissue stiffness. For that, we, for example, developed channel technology where we could clear this whole human organs and label that use a different chemistry, chops, a detergent with small micelle size that can really penetrate without getting stuck on the surface. So we do this by a chemical search that enables us to interact with tissue, permeabilize, and enables the antibodies to penetrate, to clearing solutions to penetrate. More or less, we are settled with that. I guess now we did also human organs. Now a lot of effort goes on the AI side. How do we analyze? Now, whatever we see, even the background is important information. How do we analyze that? So we are training AI for this.
Yeah, I've got a microphone. Yeah. Dr. Ali, thank you for your talk. It's very interesting. Just going back to your skull marrow studies, because you actually see the difference in the bone structures, right? With for example, your other femur, etc. Now, have you actually thought about that because of the cell origin? Because I think you're looking at the skull bones that are of neurocrest origin. So as a result of that, you may want to follow that track, looking at other tissues and cells of the neurocrest origin. It's just a thought.
In the skull marrow?
Yeah, in the skull, the skull's bones, their origin are different embryologically.
Yeah.
So I was just thinking because, there was one picture where you're looking, the bone actually doesn't cover the occipital bone. So I was thinking because of maybe it's the neurocrest cells that are, because developmentally they're different. So I'm just sort of just thinking back whether, you're following the track of neurocrest cells and origins like your adrenal cortical cells and all the
Yes.
other cells, sort of looking at that, whether, that would be, something
Reason.
of interest.
Probably it's the question is interesting. Probably it's also true that this contributes to differences we see at the molecular level.
Yeah.
As we are focused on diseases, stroke, dementia at the adult, so we are therefore, not trying to entangle the progression through the development. And there is Alain Chadotal in France. He's doing human embryo transparency and labeling, and actually he published an atlas on that, how the cranium develops over time. They did some molecular labeling too, but not in depth like proteomics or omics. They're more biased targeted antibody labeling. Probably it would be also relevant for us to understand exactly the differentiation of the cells in the skull marrow, bone marrow, how they might be interacting with brain tissue, and how we can stop them, right? We need to understand them very carefully compared to other
because
cells.
their interaction will be different.
Potentially very much,
so,
yes.
So that's why, just a thought.
Yes, good point, thank you.
Thank you very much.
Hi, beautiful work. So I'm just wondering, could you speculate how does spike protein drive long-term COVID at molecular level?
Yes, so speculation is we can speculate. I guess we don't have much data. Spike protein is highly immunogenic, right? So therefore the vaccines, they work. If you take the spike protein express, there is an immune trigger immediately. And of course, if you have this immune trigger at extreme levels during infection of shedding the spike protein, it's not good. A lot of inflammation. And also ACE2 receptor, they are also on endothelium of the vessels, right? You can see a lot of vascular targeting. And that, again, collecting, it probably induced some signaling there, which isn't good, excessive. So overall, clearly having a lot of spike protein binding and activating things in the body is not good. That is actually not probably surprising. Surprising part, at least what we reveal, is that spike protein still is in the body after years of infection. So that's the problem. So that gives us the link to long COVID, right? Long COVID could be, I think, hard to describe, but I think the simple way is to say someone suffers from SARS-CoV-2 impacts over the long term. But this could be because they got the infection very heavy and their systems are impacted, they suffer, and or there is still something bothering in the body besides this initial infection that keeps them not to recover. And that's what we are thinking is one, at least leg of the problem, this lingering spike protein. And we are designing now some peptides to, some binders that will attack them, and then immune cells will clear them. We will try to reduce the load and see whether we can reverse it.
Excellent research. I have two naive questions.
Thank you.
The first one, I didn't quite get for the Omicron and the original virus infection to the mouse. Do you use the virus or just the spike protein?
We did both. We started with spike early, and then later experiments, they have also the virus. So we have both virus, and then we use full virus and did the experiments in S3 facilities.
I see. So what makes the Omicron variant of the virus be able to spread all over the body, but not the original version?
I think a lot of studies also investigated this spike protein mutation, right, in different versions, and clearly it changes its conformation and binding feature. Now it can go and bind to new targets. It was more specific to maybe the lung tissue, then it became less specific and could bind now to entire body tissues. So it's really changing these mutations. They change their conformation that they can target much more cells throughout the body. That's what's happening, what we see at least in these imaging experiments.
And also for this inflammation of the Alzheimer patient in the skull.
Yes.
So is it initiated from the brain and somehow this inflammation is spread out to the skull, or is somehow already initiated from the skull themselves? So where is the stress or the inflammation originated?
Good question. We don't know this. We don't have the answer. I think neuroscientists would argue that it comes from the brain or inflammation starts in the brain. Immunologists would maybe say other way around. I don't know. So let's try to at least tackle whether inflammation in the skull is probably earlier than inflammation in the brain. Let's see, we're going back. So actually, I didn't mention, there is a hint in the image on that. Where are we? Somewhere here, yes. Okay. So what we see is something a bit consistent. The amount of signal, at least with TSP or PET, what we see is stronger in the skull marrow. This is a tracer developed for brain imaging, but at least in the inflammation case, you can see it's much more stronger. So what we tried to do actually in this study, but we didn't have enough patient samples, if you go earlier stages of AD, where maybe TSP PET doesn't show something in the brain tissue, would we see it in the skull marrow? I really wanted to answer this question, but we didn't have, because such an early stage, they don't go and get TSP or PET, so we didn't have. But from this, potentially one could speculate it might be that skull, at least maybe the signal is amplified, that it shows very quickly the brain inflammation. So it could be just mirroring what's happening instead of just driving it. But even mirroring is good because then you can potentially do maybe early diagnostics because the inflammation signal is amplified in the skull marrow niches. One thing that we were interested in, which we are still but didn't really started, trying to develop devices that you can put and then it kind of do local inflammation monitoring, and maybe it can give us readout of brain inflammation if there would be a disease or if you have a patient and treating with approved AD drug or how it changes. So, but of course, there isn't really a technology that you can do this, like because you cannot just put people all the time into PET, but a device that would be attached. We thought a bit ultrasound, like kind of devices, optoacoustic, photoacoustic. It needs a lot of R&D, but I think something like this can be done if any of you are engineer and wants to do some interesting project. Here is an idea.
For the AD brain, do you also have that amyloid or PET or tau PET imaging to see if that's correlated with pathology?
Okay, so for AD brains, in this case, whether we have a beta or tau imaging, we don't have. But I asked this question to our radiology folks doing all this thing. There wasn't really clear information. We didn't, I think either from the samples we have, there wasn't such a correlation or indication of, and also probably we didn't have enough scans to correlate everything. But at least initially, it didn't look as promising as this.
So those the signal that light up in the skull marrow. Mm-hmm. What cells do you think those are? Like macrophage, monocytes, or what are they?
Yeah, good question. The tracer is not very specific. This tracer, it will potentially taken up by any immune cells, even anything phagocytotic, right? We didn't do additional work on that. What could be? I mean, what we did is that rolling back prior figures in the paper, we did transcriptomic studies a lot of transcriptomics from skull with stroke. When there's stroke, things goes up. And now we compare control, sham, and then stroke skull marrow niches. And we look at the cell populations, and you can see all these graphs in the paper, the plots. So what we see is a lot of neutrophils. They seem to be, neutrophils seems to be very, first they are the number one population, and second, when we look at which cells profile changes the most, again, those are the neutrophils. Transcriptomic-wise, their profile, they change the most. So they are probably one set of the reason here, probably or at least one, we need to pay attention to neutrophils. Potentially they are changing, and that means they are interacting with tissue a lot, because we also see that they get into the brain through these channels. That we had also a bit of a work in this direction to stop their migration. So that's something, of course we got interested on the way, all these findings, but we dropped because we have to focus on what we want to do. We are a bit more at the technology side, revealing this kind of information. Hopefully people who want to dive into biology, they can do it. But a lot of interesting, I think, biology there to be explored. What kind of cells are those? What are some molecular targets that we can interact to reduce this inflammation in the skull marrow niches, whether that will have any impact on the brain inflammation or not. So really interesting questions to follow.
Thank you for the wonderful talk. And first, I really like your, I really have a great thank to you to show me about the transparent animal, because it's first time to see me, and it looks like really like a alien in the Star Wars, and that's why I like it maybe. And then I have two questions. The first question is about the SMC you already mentioned about, and you say that some of the bone marrow immune cells, some of bone marrow immune cell goes to the SMC, and the others is just go another. And is there some like distinct difference between the two groups?
So overall, we say skull meningeal connections. Then see this connection opens to the bone marrow and then goes down. Probably there are many types we measured. We did some categorization of their diameters and length, and we also sampled different human calvaria regions, frontal, side, and back. There's some density differences. With aging, it's also changing a bit. So there are many probably differences, local differences to be explored. And could that mean anything local, right? Is there more inflammation in the cortical regions versus these cortical regions or the others? We did also a bit structural analysis. So is this, when we talk about the skull meningeal connections, is this a vessel? Is it a bone structure? What is it, right? So we did some EM studies and immunolabeling. We see they have some fibroblast lining in, a bit mixed. They're also calcified, but also there is some muscular features. Some cells, they kind of protect these niches. What we know for sure, they are there. Exactly how their structural composition, how different they are at different regions, and so on, to be explored. But they are there, and they mediate infiltration of immune cells. Very interesting. We see repetitively also Johnny Kipnis, Marco Colonna, and others now. A lot of people are working on this biology now. They all see, right, in independent experiments, that there is these channels and migration. But I think another interesting thing is the other way around. So things are coming out to the brain, to these cells. Why? Why that should happen? So it looks like there is a physiological interactions, communication, even in health. And that's, I think, very interesting biology to be explored as well.
Okay, let me extend this question. And so some of bone marrows, some of the immune cells in bone marrow goes to the SMC, isn't it? and the destination is settled before they release, before they move?
I don't know.
Yeah.
We
Okay.
don't know. I mean,
Yeah.
how can we know? It's a very difficult experiment to do, right? So
Yeah.
they're now doing 2-photon imaging. We did 2-photon imaging in the paper. We could see their mobilization. We do imaging. There's a lot of cells. We do injury, and this cell starts disappearing from the field. They're going somewhere. Very crazy. But what is their destination and where do they go? We don't know. I think it would be nice to use maybe 3-photon at least. You could see a bit more to the cortex. We didn't have that technology established at that time. We don't know this answer to this question. Where do they go exactly? Which types?
Okay, the last question. So in case of the COVID-19 patient, you find out that there is some spike protein is accumulated, and can we get rid of the spike protein by therapeutic or something, or is it an irreversible reaction or something?
Say it again.
So can we get rid of the accumulated spike protein in the COVID-19 patient?
Can we read this out? Like, can we
Get rid of, yeah. Remove it.
get rid of?
Yeah.
Get rid of. We don't know. There are long COVID clinical trials in the US at the moment only, actually. So they target potentially these leftover reservoirs of virus, they think, but initial things didn't show any impact. We think it's more spike protein. That leftover is a problem. Can we get rid of it? We don't know. Probably over time it goes down, but it's not sufficient level. So we are working on it, so designing these binders, which will enable us to tag them for clearance, a bit of stealing the idea of A beta clearance and these kind of things. Hopefully, I guess we all will have impact of it in our lifetime. Probably, I believe we lost some years of our life to this disease, this infection, because seeing all this body impact targeted by this spike protein, which is triggering a lot of inflammation, this cannot be good. So hopefully we find something that will enable us to get rid of this spike protein.
Thank you so much.
No more questions. I'll leave again for one minute.
Thank you.